Good afternoon, ladies and gentlemen. Welcome to Carestream Molecular Imaging's presentation using 7 imaging modalities for efficient and accurate evaluation of new probes and biomarkers in small animal models. Due to the number of people here today, we have muted all attendees. If you have any questions, you can type them into the question box to your right. We will answer them at the end of the talk, and if we run out of time, we'll make sure to contact you directly. Without any further ado, I am now pleased to introduce you to today's speaker, Dr. Seth Gammon, our Worldwide in Vivo Product Manager here at Carestream Molecular Imaging.
Hello, everybody, and welcome to our webinar today. The plan is for this talk to run around 20 to 25 minutes, and then we'll be willing to take questions at the end. So let's get started. First, I want to introduce who Carestream Molecular Imaging is, in case you're unfamiliar with the company. And then I want to go into why multimodal imaging using 7 imaging modality actually matters to one's research. And then last but not least, I want to give several examples of how you can utilize multimodal imaging to more efficiently develop probes and biomarkers in small animal models, particularly mice and rats. Carestream Molecular Imaging is the only company that's out there that can offer our preclinical researchers 7 imaging modalities in just 2 instruments. With the Vivo MSFX Pro, researchers can image their mice or rats using fluorescence, luminescence, radio-isotopic, and radiographic, and with the Albira system, PET, SPECT, and CT. Carestream was actually recently given a very nice award for offering this product offering for our research customers, the breadth and depth of research that our customers can do with this product offering, and that was Frost & Sullivan's 2011 Best Practice Award. Frost & Sullivan is an independent research analyst group. They analyze hundreds of industries and looked at all of the competitors in the in vivo space and selected Carestream as this award winner. Why does one source matter? Why shouldn't you just go and cherry pick products from different companies? There's some very practical reasons why it's a good idea to partner with Carestream Molecular Imaging to get these 7 imaging modalities. First, for new core facilities, you can really go from having no in vivo molecular imaging equipment to having a nearly complete core facility in one purchase package with 2 instruments. That means you'd have one point of contact for application support, technical support, and service. If something were to go wrong, you don't have to reach out to multiple different vendors and organize their schedules. You'd have one continuous point of contact that you could always get in touch with and count on to get your instruments back up and operating at peak performance. By having 7 imaging modalities in 2 systems, you also minimize the total cost of ownership because within these 2 very small footprints, you get 7 modalities. We know lab space is very expensive and people are trying to get more for less, particularly now. And by minimizing the amount of floor space you either have to take up with the instrumentation or build for a new room, you can really save money in the long run, lowering your total cost of ownership. So the next question is, from a scientific perspective, why does multimodal imaging matter? I think it's first a good idea to take a step back and look at the necessary steps that one has to do to validate a new probe or biomarker before you taking it to publication or to translation to the clinic. First, you have to identify your target. This is typically done in collaboration with a proteomics or a genomics group who identifies an interesting gene or protein that's found in your cells of interest that ideally aren't found anywhere else in the body, or at least aren't present during normal development at the time you want to track those cells. The next step is working with or having your own in-house chemistry group in order to design that probe or biomarker. This is particularly critical if you have an injectable probe that you're trying to build. If you're looking at developing preclinical imaging reporters, you may not need access to a peptide or small molecule synthetic chemist, but rather would want to collaborate with a molecular biologist and biochemist who could potentially create fusion proteins and reporters for you. You then have to take those new probes and biomarkers and rigorously validate them in vitro in well-controlled assays. These typically take the form of membrane binding assays if you're looking at a receptor, or if you're looking at a protein activity, purified protein that's either commercially available or purified through your in-house biochemistry group. Once you have robust signal to noise and specificity using these in vitro assays, you then want to layer on the additional complexity of testing these in living cells or tissues outside of an animal. These would typically be assays found in 96-well plates or 6-well plates, where you're incubating the cells of interest with your probe, if it's an injectable probe, or testing the reporter in live cells if it's a genetically encoded reporter. You then want to validate the specificity against a variety of uninteresting cell types or cell types that represent normal tissue or normal biology, as normal as you can get it in an in vitro setting. The next step would then be to take these probes or biomarkers and put them into small animal models, whether it's a tumor xenograft, for example, with your target tumor, either placed subcutaneously or orthotopically placed for more relevant models. The next step is to, once you've validated specificity and targeting in vivo, you want to continually validate that target ex vivo, so looking at the key time points, extracting tissue samples, checking it by microscopy or receptor binding assays again to make sure that your target is really, your ligand is still really binding the proper target. Then, particularly if you're interested in moving to clinical translation, you want to move to larger animals, non-human primates. This might be dog models, rabbit models, monkey models. And then last but not least, moving that probe or biomarker into test in patients. Now, if you're working in the preclinical space and you're a researcher and you're really just trying to help develop new probes and reporters to help fellow researchers, you may not have to actually go any further than step 6. There's a lot of critical controls and information that you want to get out of each of these steps. The biggest thing that's critical is understanding the specificity of your probe. Everyone would love to have a probe that's 100% specific to, for example, binding only to tumors or even a subclass of tumors. The reality is that's not usually the case, and so it's important to understand what your binding specificity really is and what you can expect as false positives and false negatives when you go in vivo animal models or into patients. And this can be tested in a variety of ways. Typically, you can utilize cells or cell lines that normally lack your receptor of interest. You can make genetic knockouts or knockdowns quite readily now using shRNA or siRNA technology. If you're doing injectable probes, you of course want to do classic cold blocking or displacement studies with a competitive or non-competitive inhibitor. And an easy one is to just inject your label alone if you're looking at a fluorophore or radio tracer to make sure that the tumor, if you're looking at tumors, isn't just non-specifically taking up your label. You then want to validate how good it is. Are you getting appropriate signal-to-noise ratios versus clearance organs or other irrelevant tissues? And if you're actually looking at a clinically translatable probe, really looking hard at its predictive value by generating receiver-operator curves. Last but certainly not least, you want to look at the timing and dosimetry. Those are very much interrelated processes. You want to look, if it's a radio tracer, at how much dose a target organ or clearance organs are receiving based on the signal-to-noise ratio you need to have reasonable images. You also want to make sure that the pharmacokinetic profile of your probe or biomarker matches the kinetics of the radio tracer. With fluorescence, this isn't as much of a problem because these probes don't lose their activity over time unless they're chemically modified. So given all of the steps that really need to be taken and all the types of studies that need to be done to really drive a probe or biomarker from inception to either use in small animals and adoption by the research community or clinical trials, it's critically important to most efficiently to use your time as efficiently as possible. That's why we believe having an MSFX Pro and an Albira and these 7 imaging modalities allows you to drive your research forward by as quickly as possible, narrowing the funnel of your probes that are moving from inception to the animal or patient. And you want to narrow that funnel as quickly as possible, whether you're in academia or pharma and biotech. So, as I mentioned before, having live cell or in vitro assays is critical. The nice thing about the in vivo MSFX Pro is that while it's primarily used for whole animal imaging, it's certainly available and can be used for your in vitro assays like Western blots. It'll be important for looking at protein expression levels, or whole animal, or whole cell binding assays. We have an in vivo MSFX Pro is a great tool then to take any candidate that makes it through your in vitro and live cell assays and moving it forward to small animal imaging. With fluorescence imaging, it's much more cost effective on a per assay basis to study these probes and biomarkers and rapidly validate up to 5 animals at a time the binding specificity of your new probe or biomarker. Once you've validated that in fact you have a probe that has good specificity, that's blockable and doesn't bind to your negative control cell lines, for example, then you really want to understand the quantitative pharmacokinetics of those probes. Then you really want to move to a truly quantitative 3D molecular imaging system like the Albira PET SPECT or CT. Particularly with a PET CT, you can quantify and model the uptake and binding of those probes down to really the nanomole per liter level. And then once you've rigorously validated that you have something that doesn't bind to non-target organs, you have an understanding of what the clearance profiles are and does it have a chance of being safe in humans due to dosimetry issues, you can think about moving that onto your clinical imaging or clinical trials. Of course, that's a fairly simplistic model. At every one of these steps, you're really going to want to take information based on what you learn from, say, a PET experiment and go back perhaps to optical and rescreen your biomarker. You may learn that you need to change its clearance profile, and so it might be much more efficient to check it with optical after you modify it by, let's say you need to shift it from a liver to kidney distribution as you make it more hydrophilic. It'd be a fast way to just check that you shift it from liver to kidney and optical before going back forward and redoing your quantitative PET CT studies. Each of these levels have their own pros and cons, so that's why we do them. I just want to go through them very briefly here. For in vitro assays, the pros are obviously that we can have very precisely controlled experiments. We know exactly how much probe we've put in. We know how much ligand is present, how many cells are present. We should be able to control nearly every aspect of what's going in. The problem is it's a very artificial environment. As you move to live cells, you get away from that, but you also lose control over things like receptor enzyme expression levels. Optical imaging at the next step is very precise, it's very cost effective, and frankly much faster to acquire the images as well as analyze the data. Typical acquisition times are somewhere on the 30-second to 2-minute time frames. It allows you to study your probes in the most relevant models, mice and rats, other than humans. The problem there is that it's inherently semi-quantitative. That really has nothing to do with the instrumentation and everything to do with the nature of fluorescence or luminescent probes. The ability to make activatable probes means that these probes are sensitive to their local chemical environment. And so if the brightness can change based on the local chemical environment, it's inherently semi-quantitative. One of the nice things about nuclear medicine is that a radio tracer that you put in water or radio tracer that you put in a membrane or bound to a protein, it always has the same rate of decay. If you move on to quantitative 3D, that's also self-explanatory. You get much more quantitative data, again, because of the nature of the probes. Also, with PET particularly, you can do kinetic modeling, which is something you really can't do with optical because you don't have it in the distribution in 3D. And these now then become translatable. There really isn't the instrumentation in the clinic today for in vivo optical imaging in patients, whereas there's plenty of instrumentation and know-how out there in the clinic for imaging PET SPECT probes. However, the cons are the cost and potentially probe availability and requiring a radiochemist on your site. So as you can see, by going through each of these, you really have a chance to leverage the pros at each step and then minimize the cons by going down in order. So if we started with quantitative 3D molecular imaging all the way at the top, just for hyperbole's sake, you'd be spending way more money than you need in order to validate these probes. So first, let's take a look at some examples of how people have used optical imaging to find better ways to create peptides to deliver cargoes across the blood-brain barrier. This was a group that wanted to utilize TAT peptide or TAT peptide-like protein, TAT peptide-like sequences in order to deliver cargoes across the blood-brain barrier. So rather than starting with a radio tracer or starting with a drug, they just labeled it with Psi 5.5, near-infrared fluorophore, which is where you want to work in vivo, modified that peptide with various chemical moieties, injected them and monitored them in real time until they got a formulation and an injection scheme that worked well to give them nice signal coming from what appears to be the brain. However, showing that something is in the brain isn't the same thing as showing that it's actually crossed the blood-brain barrier. One of the nice things about fluorescence that's different from luminescence or radioisotopes is that you can really zoom in and get a lot of microscopic detail and show that this has not only gotten into the brain, but in this case they stained for endothelin in green, and they were able to show that the peptide, as indicated here in red, was outside of the blood-brain barrier. Then if they wanted to take this peptide further, they already have a formulation now that they're confident goes not only to the brain but crosses the blood-brain barrier. The same group was also interested in designing probes that might more effectively target A-beta, which is a critical imaging target for many people to look at ways we can improve treatment for Alzheimer's disease or perhaps predict Alzheimer's disease early, although amyloidosis occurs in a lot of other diseases as well. To do that, they looked at an analog of curcumin, which they expected to be able to bind to A-beta, and they modified it with a difluoroborane in order to make it fluorescent, and as we'll notice later, might make it a possible PET candidate as well. First, they validated this in vitro, as we talked about. That's just good practice. So they showed that whenever they add the compound in with A-beta aggregates, they see an increase in fluorescence, and they validated that it bound to histological sections ex vivo. They then took that same probe and imaged it over time in transgenic animals versus negative control animals, versus transgenic A-beta positive animals, and they're able to see at nearly every key time point an increase in fluorescence over the control experiments. And again, as you can see, optical data can be very, very precise, even though it's semi-quantitative, and we can quite readily separate out this experimental group from the control. So we're pretty sure that this is actually working. In fact, they then went on to complete the workflow and did ex vivo analysis to show that that small molecule is in fact binding onto the A-beta. They went on to do additional negative controls, including ICG, to show that they're not just looking at changes in blood-brain barrier integrity. What's interesting about this particular probe is you could easily imagine labeling this with either carbon-11 or F18 at one of these two sites. The challenge is that with optical probes, you saw an increase in fluorescence, so that could have been contributing to the ability to see it over background. But at least they know that this has a shot of working quite rapidly without needing to go through any costly radiosynthesis or PET SPECT or CT experiments from the beginning. If they want to take this forward, now they could go on to the radio labeling part because they know it at least has a shot of working. If you wanted to do very simple experiments to look at things like changes in perfusion, you can, of course, utilize SPECT. There are plenty of probes that are out there that do that. If a similar group wanted to monitor changes in blood flow in the Alzheimer's versus normal mice and confirm they were really seeing binding, they could, in fact, check that with Tec99 HMPAO to ensure that they were seeing a change in an increase in uptake that was really due to binding to their target, not just changes in perfusion. Looking at a similar model, another way to look at molecular imaging is how can we take existing probes and apply them to new problems. And F18FDG is an FDA-approved molecular imaging tracer, and so people are looking at how, across many different models, how can we apply this understanding. And this group looked at how does, this is from Dr. Pozo's lab in Madrid, use the Albira to look at what are the changes in metabolism that we see with Alzheimer's. And you can quite readily see that in transgenic mice that are Alzheimer's-like, you get significant decreases in metabolism found in the brain by FDG. Now, that might mean we could take that forward to the clinic, but certainly there are a lot of ways that metabolism changes in the brain that doesn't have anything to do with Alzheimer's. However, an imaging lab that wanted to look for ways to develop treatments to improve the outcome of Alzheimer's patients could easily utilize this particular probe in a preclinical setting where they wanted to reverse this APP phenotype of decreased metabolism back to control. Here's another example of how you could look at metabolism from a preclinical perspective and try and reverse the effects of, for example, aging. So as we mentioned, Alzheimer's is one reason that glucose consumption could be decreased, but another reason could just be that as we age, our metabolism decreases. And in fact, that's what was seen in this study. Again, the nice thing here is we're utilizing a already available probe, and then researchers can utilize this biomarker to develop strategies to reverse this decrease in metabolism in models of aging. Another group are interested in developing probes for looking at microbial surfaces. They have a very general platform that is a reporter coupled to a linker, coupled to dipicolyl amines and zinc. These are known to bind anionic phospholipids, and bacteria have an abundance of anionic phospholipids on their surface. The reporter in this case is a fluorophore. So first thing was to validate it in vitro and show that it's still bound to the surface of bacteria, and it does. Next, they wanted to validate in an in vivo model of infection where they injected Staph aureus into one leg and then into the contralateral leg vehicle alone. The probe was then injected intravenously. This is not an activatable probe, so it diffuses throughout the body in an on state. Will eventually bind to the target, hopefully, and then clear away from the rest of the body, leaving a bright surface. And in fact, that was what happened. You can see the probe after injection distributes throughout the animal and then begins to be specifically retained at the target site by 12 hours. And they were able to look nicely at these target to non-target ratios to show that this is a probe that's worth pursuing. And now they're looking at taking this probe to the next level or something like this probe by labeling it with copper-64. And in fact, in very early preliminary experiments, they were able to see binding of this at an infection site. So how did optical imaging help them? This is a group that would not have had the resources or ability to get to a PET or SPECT experiment right away. And in fact, they wanted to triage compounds that they knew just fundamentally wouldn't work. And they were able to do that quite successfully and are now looking at how can they transition these molecules into PET agents for imaging infectious disease. Another big area of work, of course, is looking at new surface biomarkers for oncology. This group wanted to start from really scratch and do combinatorial synthesis of peptides that might bind new surface targets. So their strategy was to do combinatorial chemistry, label it with a fluorophore, and validate that it binds specifically using fluorescence before they think about how can they move it on to some other potential translatable strategy. So this group does a great job because they show that, okay, in a subcutaneous xenograft, my probe is binding, as you see by the increase in fluorescence at the target site. However, whenever they show a no tumor, all right, they're able to show that it's not just randomly going to the site for some other reason. That doesn't really tell us a whole lot about the specificity, though. So they wanted to show that if they just add their dye alone, that it doesn't get specifically taken up by the tumor. And in fact, you see significantly less tumor uptake here in the dye alone than you see in the tumor. That's a critical control because tumors take up a lot of fluorophores and macromolecules for random reasons that frankly we don't well understand yet. They then did a scrambled peptide sequence, which is great because it shows that it's not only specific to the sequence, to the amino acids in the sequence, but the exact order matters, and we're able to do a cold block study. So really they did a lot of the really nice biochemical validation using just optical before even needing to think about taking this on to a radio tracer type assay. They then went on to do genetic negatives, which is another great control where they take a negative background and then overexpress their target. And in fact, you can quite clearly see in the same animal, tumors receiving roughly the same dose, that you get nice specific binding to the one that overexpresses it. This is quite an elegant paper. If you want to look at a really nice set of experiments to validate these, this is the one to look at. With the in vivo MSFX Pro, in some cases you can skip straight to doing 2D radioisotopic imaging rather than going fluorescence then radio labeling. So in this case they had a peptide that they felt would target a specific tumor, and they used the 2D radioisotopic probe to pick up signal from this Tec99M labeled peptide. They showed that, just like in the other group did, they did a nice job where they showed that the tumor lights up, so they're seeing targeting. Whenever they block it with competitive inhibitor, the signal goes away. If you look at tumors that are known to be negative for this particular antigen, you also don't see any uptake. And then they can now take this forward right away to a SPECT CT system. And particularly for SPECT isotopes, and to some extent PET, having this 2D radioisotopic screen might in fact allow you to bypass fluorescently labeling them and go straight from a screening protocol using the MSFX Pro, using the exact same chemical moiety, into 3D SPECT CT. And of course, one can evaluate existing probes using the Albira for any PET or SPECT probe that's out there and been developed in the decades of research that's been going on for evaluating these probes. And you can see these not only in 2D but in quantitative 3D images. With these modalities, you can also conduct longitudinal studies just like you can conduct them with optical. Because the mice stay alive, we're not sacrificing them, and you can watch how these probes bioavailability or uptake changes over time. For in this example, you're looking at tumor growth in a mouse. And you can utilize existing biomarkers such as FDG to look at candidate therapeutics, where you have a longitudinal study, day 9 to day 10, and inject with, they wanted to look at direct injection chemotherapeutics, so they injected with saline, ethanol, or a candidate, and were able to show that they see significantly more killing with the candidate than with the ethanol. And pay attention to this small little hole of metabolism that you see. In fact, with a high-resolution PET-CT like you get with the Albira, you're able to almost do in vivo histology. So when they did the histology to confirm this change in metabolism, you see a mild damage at the core and then severely damaged edges on the candidate injection, just like you saw in the 3D image. So in summary, by having all of those imaging modalities, you can most efficiently use your time and drive your research forward as fast as possible for and for really the least amount of money possible. because you're not wasting money by starting with a quantitative 3D system if you don't really need to use it, when a high-resolution optical system could, in fact, answer a lot of your binding and specificity questions first before going on to confirm in quantitative 3D that you're seeing the targeting that you want and make sure you're not didn't miss some overlapping or deep tissue signal, particularly for rats. So in conclusion, Carestream provides you access to 7 imaging modalities, and we believe you can accelerate your research by applying the correct molecular imaging modality to the right step in the validation process, and this will help get you to the answer much faster, and particularly important for commercial biotech companies and pharma companies to reject compounds that fundamentally aren't going to work as soon as possible. And I'd like to thank you very much, and I'll be glad to take any questions.